Welcome back, guys. Now in this video, let's discuss about male reproductive physiology. We'll be discussing about different hormones and different structures in the testes, how spermatogenesis is going to happen, and all the important topics which are needed for your exams that we are going to discuss in this video. Okay, so let's begin. Male reproductive system. Okay, and the male reproductive physiology. First, I want to start from the basics. Okay, see, our reproductive system is controlled. Okay, the reproductive hormones, everything is controlled by the brain. Which part of the brain? It's the hypothalamus. So let's start from the hypothalamus. Imagine here is the hypothalamus. Okay, here is hypothalamus. Now there is this one hypothalamic nuclei. This hypothalamic nuclei is producing GnRH, gonadotropin-releasing hormone. Now this gonadotropin-releasing hormone is going to come to where? It's going to come to the anterior pituitary. So now this gonadotropin-releasing hormone is going to act on a cell in the anterior pituitary. This cell is called as gonadotrope. When the gonadotropin-releasing hormone is acting on this gonadotropes, now this gonadotrope is going to produce the gonadotropins. It's going to produce the gonadotropins. What are these gonadotropins? Gonadotropins here are FSH and LH. Okay, as I am discussing about the male, I am going to talk about the testes. Here the gonad which I am taking here is the testes, not the ovaries. So now look what is this FSH and LH are doing. Okay, look here, let's imagine here is the testis. Now in this testis, let's say there are two cells present. For easy understanding purpose, let's say here is one cell and here is the other cell. So now this LH is acting on this cell, L for L. So this LH is acting on this cell. Okay, so what this cell is going to do? When the LH is acting on these cells, the LH receptors are present on these cells. When the LH is acting on these cells are producing testosterone. Okay, testosterone, which is a steroid hormone, testosterone. So what is this cell? This cell from which the testosterone is getting produced, this cell is called as the Leydig cell. Okay, so interstitial cell of Leydig, actually we call it as the interstitial cell of Leydig. It's the interstitial cells of Leydig which produce a testosterone. Okay, now once you have sufficient amount of testosterone, what this testosterone will do? The testosterone is going to give the negative feedback to the LH. So testosterone gives the negative feedback to LH. Now, what is the fate of this testosterone? See, that testosterone is going to, in the males, when you are a child, it helps in the development of internal genitalia. Okay, it helps in the development of internal genitalia. Now, this testosterone, what it does is it's going to convert into the more powerful testosterone with the help of enzyme called as a 5-alpha reductase. With the help of enzyme called as a very important 5-alpha reductase, the testosterone will be converted into dihydrotestosterone. This dihydrotestosterone is responsible for external genitalia development. Okay, external genitalia development, that is the penis and scrotum. For the external genitalia, you require the dihydrotestosterone, and for internal genitalia, you require the testosterone. Not only that, one more important point about this dihydrotestosterone, this dihydrotestosterone is also going to cause male pattern baldness. Okay, in the males will get the baldness, right? That male pattern baldness is because of this dihydrotestosterone. Male pattern baldness. Okay, and not only that, it causes some problems in older ages. Okay, I'm including here the pathologies. Dihydrotestosterone is also going to cause a pathology that is called as a benign hyperplasia of prostate. There is benign hyperplasia of the prostate which is going to cause a problem with the micturition. Okay, so that's a problem because of this dihydrotestosterone. In the 60 years, 70 years, in the older males, it's going to cause benign prostatic hyperplasia. Okay, now what else I want you to know here is this FSH, follicle-stimulating hormone in the males. What is it doing? The follicle-stimulating hormone, it's going to act on this cell. Okay, it's going to act on this cell. So this cell is called as a Sertoli cell. Okay, this cell is called as what? Sertoli cell. Now the Sertoli cells, when FSH is acting on the Sertoli cells, these Sertoli cells are going to make something called as inhibin B. Inhibin B. Now B for boys, remember inhibin B is produced in the boys by the Sertoli cells. Now this inhibin B, it gives negative feedback to FSH. Okay, it gives negative feedback to FSH. Not only that, what else I want you to know is this testosterone which is produced. Okay, the testosterone which is produced from the leading cell. This testosterone, some amount of testosterone, little bit testosterone will come to the Sertoli cells. The Sertoli cells will take this testosterone. Now, with the help of enzyme called as aromatase, okay, let me write it here, with the help of enzyme called as aromatase, the testosterone will be converted into estrogen. So, these Sertoli cells, remember, Sertoli cells also produce estrogens in the males. Yes, where the estrogens are coming in the males? It's the Sertoli cells which produces the estrogens. And estrogens, what is the function of estrogen? Estrogen helps in sperm development. Helps in spermatogenesis. Okay. Actually, in the sperm, you know, in the top of the sperm, there is a cone-shaped area which is called as the acrosome. For the acrosome, a biosynthesis, for the formation of that acrosome and the acrosomal enzymes, you require this estrogen. So, this estrogen, tell me what is it helping in the spermatogenesis? That too, acrosome biosynthesis. Okay, in the acrosomal biosynthesis. For the production, you require the estrogen. So, estrogen receptors are there on the sperms. Estrogen receptors are there on the sperms. They will ask you on the sperms which receptors are there. Most of the students will think sperms have the testosterone receptors. Sperms does not contain the testosterone receptors. Sperms do not contain testosterone receptors. Sperms contain estrogen receptors, and those receptors are going to help in the formation of the acrosome. Okay, and not only that, see, these estrogens, what they will do, they are going to cause the closure of very important FMG question. They cause the closure of epiphyseal plate. Okay, they cause the closure of epiphyseal plate after the puberty. Okay, so when the epiphyseal plate is closed, the growth spurt, okay, the growth spurt is going to be stopped. So who is going to stop the increase in the length of the bones? It's the estrogen. So estrogen causes the closure of epiphyseal plates, so stops growth spurt. So these are some important points which I want you to know in the male physiology. Okay, there are in the testes, there are two types of cells, Sertoli cells, Leydig cells. So the basic functions here I have explained you. Later I will be explaining most important points about the Sertoli cells, what else it secretes. Okay, so how the Sertoli cells are forming the blood-testicular barrier, how they are going to be helpful for the spermatogenesis, okay, how they are acting as a nursing cells, okay, that we will discuss later, okay, but these are the basic points which I want you to know. Next, where are the gonads present? You know, the testes, where are they present? They are present outside the abdominal cavity. They are present outside the abdominal cavity. Actually, testes are an intra-abdominal organ. When you are an embryo, testes were developed inside the abdomen. But later, they descend down with the help of a spermatic cord. These testes will descend down into the scrotum. Okay, why they are placed outside the body? Because outside the body there is less temperature. That less temperature is required for spermatogenesis. You understood? So you need to maintain the temperature. If there is more warm temperature, that's going to kill the sperms. That's not good for the sperms. Okay, so you require the less temperature for the proper production of the spermatogenesis. So that's why testes are outside the body cavity. So in the scrotum, the temperature is somewhere around 32 degrees to 35 degrees centigrade. So that's the optimal temperature of the spermatogenesis. That's the optimal temperature. That's a good temperature. Okay. Now, how the temperature is regulated? How this heat is regulated? For example, let's say, imagine here is the, let's draw the scrotum. Let me show you the scrotum. See, if this is the scrotum, okay. Now here are the testes. Okay, here is one testis. Here is the other testis. Let me show you. Actually, the blood is going to come to the testis, right? The blood is going to come. From where the blood is coming? The blood is coming from the body. Okay, artery. See, let's say artery which is bringing the blood like this. A testicular artery, which is a branch of abdominal aorta, you know it. Okay, it's bringing the blood. I am drawing a easy diagram so that you will understand. See, imagine this is the testicular artery. Now, testicular artery, it is bringing what blood? It is bringing warm blood. It is bringing warm blood. So, this warm blood may increase the temperature inside the scrotum. That is not a good thing for the spermatogenesis. So, that's why you have to regulate. You have to lose this heat. How that actually happens, you know? See, here there is a plexus of veins. Okay, there is a network of veins for easy understanding purpose. Look, the blood is going in this direction. In the testicular artery, the blood is going in this direction. Actually, the warm blood is going into the testis, which is not good. So, you have to remove the temperature. So, that's why, do you know what happens? Here, there is a plexus of veins. The venous blood is going in the opposite direction, from down to up. Okay. So, let me show you. What are these structure? What is this network of blood vessels that I am showing here? This is called as a pampiniform plexus of veins. Okay, these are pampiniform plexus of the veins. Okay, what they will do? They are just near to the testicular artery. Okay, they are just near to the testicular artery. So, testicular artery is trying to bring the warm blood into the testis, but this venous blood which is going back, it will exchange the heat. Okay, now you are keeping these two blood vessels side by side. In one blood vessel there is warm blood, in the other blood vessel there is normal, light, cold blood, let's say. Now, you know, heat is going to move from the temperature will move from high temperature to low temperature. So, these pampiniform plexus of veins, venous blood, cool blood is going to absorb all the heat. So, there will be heat transfer. So, let's say here, let me show you one way in an easy way. Here, the warm blood is moving in this direction. Here, the cold blood is moving in this direction. So, what is happening? There is heat transfer, heat exchange. So, this is called as counter current exchange. Counter current heat exchange to maintain the temperature in the scrotum, so that will be helpful for the spermatogenesis. So, this is the point which I want you to know. After this, I want you to know the structure of the testis. Okay, first look at here. See, now you know here is the testis. If you do the cross section of the testis, inside the testis, are you able to appreciate these small tubules? So, these tubules are called as what? Seminiferous tubules. This is the place where, okay, these are tubules, these seminiferous tubules, see, they are connected, these seminiferous tubules, they are connected to the rete testis. Okay, this area, this plexus which I am showing you, okay, so this area which I have marked right now, okay, let me mark it. So, this area, okay, so that is called as a rete testis. I have explained you seminiferous tubules. The seminiferous tubules are the places. Okay, imagine this is a seminiferous tubule. This is a seminiferous tubule, the place where sperms are going to develop. Seminiferous tubules are the place where spermatogonia are present. Those spermatogonia will be undergoing meiosis 1, meiosis 2. Later, sperms are going to be produced in the seminiferous tubules. So, this is the place where your sperms are getting produced. Okay, the seminiferous tubules, all of them, they are connecting into rete testis. All this rete testis, see, the rete testis, from here, the sperms will move to where? The sperms now will move to this area which is called as epididymis. Okay, so this is called as what? Epididymis. Epididymis, there is a head, there is a body, there is a tail. Okay, head, body, tail, epididymis. So, tell me, inside the testis, you are having seminiferous tubules in the place where sperms are going to get developed. Seminiferous tubules are going to fuse and form the rete testis. Okay, they are all coming and forming the rete testis. Rete testis is going to drain into epididymis. Now, this epididymis, this is the tail, right? This epididymis, it continues as vas deferens. From the down, it continues as vas deferens. So, the sperms are going through the vas deferens. Okay. So, this is the sperm pathway. From the vas deferens, it will go into, later it will go into the ureter. They will go, like, not into the ureter, sorry, they will go, they will just wound around the ureter, and they will go and fuse with the urethra, and from there it is going to come out. Okay, sperms are going to come out through the penis. So, now, what are the points which I want you to know here is, now, let's make a cross section here. Okay, now imagine, let's make a cross section here at the level of seminiferous tubule. Let's make a cross section here like this. Now, when we make a cross section, what you are going to get? See, you are going to get the seminiferous tubule. Here I have already shown you. Okay, here you are going to get the seminiferous tubules inside the testis. Okay. Now, inside the seminiferous tubules, what's going to develop? Sperms are going to develop. Sperms. So, this is the place where sperms will develop. See, here I am showing you already. This is the one seminiferous tubule. Inside the seminiferous tubules, you should appreciate that here are the sperms present. Okay, here are the sperms. Now, in between the seminiferous tubules, okay, outside I should say, here, these cells are there. Okay, there are these cells. These cells, these blue color cells which I am showing you here, these are called as interstitial cells. Interstitial cells of Leydig. Okay, interstitial cells of Leydig. Now, these interstitial cells of Leydig are the ones MCQ in your exam. These interstitial cells I have told you, these are the ones which produce testosterone. They produce the testosterone. Okay, it's the interstitial cells, not the seminiferous tubules. Interstitial cells of Leydig which is going to produce the testosterone that gives the negative feedback to LH. Okay, so these are the points which I want you to know. Now, after this, here I want you to concentrate on one seminiferous tubule. Okay, imagine here is the one seminiferous tubule. Let's say this is the one seminiferous tubule. I am taking this one area. Okay, I am just zooming into this one area. Okay, from periphery to center. Okay, from the periphery to the center, I am just taking this and I am just zooming it. So, it's going to look like this. Look. So, this is the boundary. This is the boundary of the seminiferous tubule. It's actually like this. It's actually like this. So, now you understood. This is a seminiferous tubule. I am taking this small area. Okay, here also it was very clearly given. Look, here I am taking this part of the seminiferous tubule and I am just showing you. Now, this area is called as the basal lamina. This area is the basal lamina. Okay, basal lamina. Now, are you able to appreciate within the seminiferous tubule? In the seminiferous tubule, there is this Sertoli cell present. Okay, here is the Sertoli cells. Actually, this whole pink thing. The whole pink thing is a Sertoli cell. The whole pink thing. Okay, this is the Sertoli cell. Okay, this is the Sertoli cell. Now, Sertoli cells, where are they present? Sertoli cells are present within the seminiferous tubule. What the Sertoli cells are doing? So, remember Sertoli cells as a nursing cells. They are like nurses. Okay, they give proper nutrition to the sperms. They are the ones which are going to take care about the process of spermatogenesis from the starting to the end. From the starting to the end, it is the Sertoli cells. As far as Sertoli cells helps in spermatogenesis continuously, every stage. For example, look here. Here is a spermatogonia. Okay, primitive cells, spermatogonia, primitive spermatogonia. Okay, later the spermatogonia will be converted into spermatocytes, primary spermatocyte, secondary spermatocyte, later into spermatids. Spermatids will be converted into spermatozoa. Okay, so actually from the periphery to the center. Okay, from the periphery, in the periphery there are primitive cells present, spermatogonia present, and when you are going into the center, there is more maturation. Okay, there is maturation. Spermatogonia is getting converted into spermatocyte, primary spermatocyte, secondary spermatocyte, spermatids. Spermatids will be later converted into spermatozoa, the mature sperms. So, from the periphery, the primitive cells are getting converted into step by step, they are converted into mature cells. So, in the center you will see spermatozoa. That's why even in this cross section, in the center in the center area, you are seeing this matured cells, spermatozoa. Okay, let me erase it. It's in the center, you can see the spermatozoa in the center. Now, what else I have to teach you? Some important points about the Sertoli cells, very important. Sertoli cells I have told you, nursing cells. Look here carefully. Here I am showing you the cross section of the testis. Okay, the cross section of the testis. In the cross section of the testis, you know here what is this circle? This circle is one seminiferous tubule. This is the seminiferous tubule. This is the center. Okay, I have told you. Say, what is this cell? What is this cell? Okay. This cell is nothing but your Sertoli cell. Okay, this is your Sertoli cell. Now, what these Sertoli cells are doing? These Sertoli cells, see here, all these are the Sertoli cells. These yellow color cells which I am pointing here, these are all the Sertoli cells. These Sertoli cells, they are helping in the process of spermatogenesis. So that's why in the center, look here, all the center, these are the mature spermatozoa. They are all having these tails. Okay, they are all having these tails. See, this is one, look here, this is one Sertoli cell. Here is the other Sertoli cell. Look, okay, Sertoli cells. Now these Sertoli cells, see what they are doing, they are hugging. Okay, they are just holding the primitive spermatogonium. This is the spermatogonium. Here are the primary spermatocytes. This is the secondary spermatocyte. Here is a spermatid. Here is a spermatozoa. Means you look, appreciate the image that these Sertoli cells, they are having these processes. Okay, they are having these processes and they are encompassing totally. They are causing the enclosure. Okay, around the different stages of the spermatogonia and the spermatocytes, so helping in the process of spermatogenesis. Now, I want you to know a few more important points here. Okay, a few more important points here. Look at this. In this diagram, it is clearly given. Look, here this is one Sertoli cell. You know, the same thing. The same thing. Here is the basal lamina. Okay, this is the basal lamina. Let's say this is the basal lamina. Okay, the periphery of a seminiferous tubule. Okay, the periphery of the seminiferous tubule, this is the basal lamina. Now, look, this is the center. Okay, here is the center where you are going to see the mature spermatozoa. Now, look at the Sertoli cell. See, here is one Sertoli cell. Here is the other Sertoli cell. These Sertoli cells, in between them, see, you are having the spermatogonia, spermatocyte, all this. But between the Sertoli cell, one Sertoli cell and another Sertoli cell, they are having these extensions, and in between this extension there is a tight junction present. Okay, so the point which I want you to know is one Sertoli cell is going to tightly bind with the other Sertoli cells. Okay, so let's say like this. If this is the seminiferous tubule, okay, here is a seminiferous tubule. Here is one Sertoli cell. Let's say like this. One Sertoli cell. Here is the other Sertoli cell. Other Sertoli cell. Actually, they are having projections, but for easy understanding, I am showing you. These are the Sertoli cells. Okay. Now, one Sertoli cell is having one extension. Here is having one extension. These extension, in between these extensions, what is there? There is a tight junction present. There is a tight junction. Okay. So let me continue this so that you will understand better. So here is one Sertoli cell extension. Okay. So in between these extensions, what is there? There is a tight junction present. Now, because of these tight junctions, look, now these Sertoli cells, they are forming a barrier. Okay, they are forming a gate-like thing. They are forming a barrier. So now no toxic substance can directly enter here. So into the seminiferous tubule, you can't enter into the seminiferous tubule because these Sertoli cells, they are forming a barrier with the help of tight junctions between them. So the junctions between the two Sertoli cells is so tight, nothing can escape into the seminiferous tubule. Toxic substances cannot escape from the blood into the seminiferous tubule. So the point which I want you to know here is these Sertoli cell, this is the Sertoli cell, it forms blood-testicular barrier. Okay, they forms blood-testicular barrier. So if there is some toxic substance in your blood, for example some drug or some some toxic substance, they can't directly get access into your seminiferous tubules because that's a place where your future generations are resting. That's a place very important for your future generations. If there is any toxic substance or a drug or a chemical that can cause the mutations, so we don't want that. Okay, so tell me, blood-testicular barrier is formed by which cells? Sertoli cells. Inhibin B is produced by Sertoli cells. Nursing cells are Sertoli cells. Spermatogenesis helping cells are Sertoli cells. All these are the important points. Okay. Now, one more important point. Look at here. Because of this barrier, okay, now you are saying there is a barrier. Okay, Sertoli cells are forming what? Barrier. Because of it, the entire seminiferous tubule, it is actually divided into two compartments. Here is the compartment number 1 outside in the periphery. Here is the compartment number 2 inside. There is outside compartment, there is inside compartment. The outside compartment is a place where here you are having spermatogonia. Okay, here you will be having what? Spermatogonia, primitive spermatogonia will be present here. Okay. and this is the area, the central area is the place where the sperms are getting differentiated, spermatogonia into spermatocyte, spermatocyte to spermatid to spermatozoa. Okay, so this second compartment is the place where actually the sperms are getting mature. Okay, that you can understand. So what are these two compartments? See, in this diagram, again come to this diagram, it was very clearly given. See, here the compartments were given. Okay, so this outside compartment is called as the basal compartment. Okay, see this compartment. Okay, this is the basal compartment. So basal compartment, what is there? This spermatogonia are present. Who is present in the basal compartment? Spermatogonium. And what is the second compartment? The second compartment is called as the adluminal compartment. Okay, adluminal compartment. So in the adluminal compartment, that's the place where actually spermatogenesis is happening. The spermatogonia are getting converted to spermatocyte, the spermatocytes into spermatids, and spermatids into spermatozoa. So how many compartments are there? Tell me. There are two compartments. What are they? One is the basal compartment. Okay, basal compartment. And second one is adluminal compartment. Now tell me, who is outside the blood-brain barrier? Who is not blood-brain barrier, blood-testicular barrier? Who is outside the blood-testicular barrier? The spermatogonia. The spermatogonia are actually located outside the blood-testicular barrier. They are outside the blood-testicular barrier. They are present in the basal compartment. So right. So outside blood-testicular barrier. Who is there? Spermatogonia. Spermatogonia, these are the cells which are present outside the blood-brain barrier. Sorry, blood-testicular barrier. And don't forget, okay, blood-testicular barrier is formed by which cells? Sertoli cells. Okay, Sertoli cells along with the gap junctions. Okay, along with the gap junctions, Sertoli cells are forming the blood-testicular barrier. Okay, the same thing. Here I am showing you a better image. Okay, a better image. See here, what is this thing which I am highlighting here? This is the basal lamina. This is the periphery of the seminiferous tubule. That's the periphery. Now, what are these things? These are the spermatogonia. These are the spermatogonia. Okay, I have told you. See here, what is this? This is one Sertoli cell. This is actually Sertoli cell. Okay, that's the Sertoli cell with these extensions. So it's forming, see, these extensions. Okay, so with these extensions, what is it forming? It is forming two compartments. Okay, it's forming the two compartments. So let me highlight the two compartments here for you. So here is the first compartment. Say for example, let's say like this. Okay, so this is the first compartment, that is the basal compartment. Here is the second compartment, that is the adluminal compartment. In the center, the sperms are getting matured. Okay, in the center, the sperms are getting matured from the spermatids to spermatozoa. Now, some important points which I want you to know. In between these two compartments, okay, there is a basal compartment and there is a adluminal compartment. Which is more important? You just tell me. It's the adluminal compartment which is more important. So, in the adluminal compartment, they will ask you which hormone levels are maybe more. Where do you think more testosterone will be available? Testosterone is helping in the process of spermatogenesis. Where do you require it? In the basal compartment or adluminal compartment? In the adluminal compartment. So, testosterone levels and estrogen levels for the process of acrosome biosynthesis. So, estrogen levels, okay, testosterone levels, potassium levels, they will be more in the adluminal compartment. Their levels important. So, potassium levels, testosterone levels and estrogen levels are all going to be more in which area? The adluminal compartment. Okay. So, after this, what else I want you to know? Any other important points about the Sertoli cells? Okay, let me write here. Sertoli cells. Let's write all one by one. They forms blood-testicular barrier, MCQ. Okay. Second, which cells are protecting the adluminal compartment? It's the Sertoli cells from the toxins from the blood. the toxins from the blood are not able to enter into the abdominal compartment. So who is protecting that? Sertoli cells. So protect abdominal compartment. Okay. So next, spermatogenesis. Okay. Third function is helps in spermatogenesis. How? Because they are the nursing cells. They give the nutrition to the sperms. So nursing. They are the nursing cells. Not only that, look, they are forming this blood testicular barrier. So immune cells, they can't get access into the seminiferous tubules actually, okay, or the antigens whatever are there within the seminiferous tubules, okay, the antigenic material whatever is there in the seminiferous tubules, they are not exposed to the immune system actually, okay, the antigenic material whatever is there in the seminiferous tubules, if there is any antigenic material, usually our immune system is not knowing about the antigenic information in the seminiferous tubule, okay, the seminiferous tubules are a highly private area. The immune cells don't get access over there, so they don't have any information about what kind of material is there. So now look, if this, for example, let's say, if the antigenic material, for example, some sperm component or something, okay, if the antigenic material, if it is leaked into the blood, what your immune system will do? Immune system is going to recognize it, and they don't know about this information. So your immune system is going to produce the antigen, your immune system is going to produce the antibodies against it. So that may trigger the autoimmune diseases. Okay, that may trigger the autoimmune diseases. So what I am trying to put into your mind is, guys, look, imagine this is the seminiferous tubule. Okay, and this is a highly private area because what is there? There is a blood testicular barrier formed by the Sertoli cells. So this antigenic material, whatever is there here, your immune system doesn't know about this information. Okay, actually these are your own self things. Okay, your own material, your own proteins, your own antigens, but your immune system doesn't have any access about these antigens. Okay, so for example, say, imagine if this blood testicular barrier is damaged. If the blood testicular barrier is damaged means now this antigenic material, it will come into the blood, so that will trigger the immune response. Now that will cause the antibody production. Those antibodies will come and attack the antigen sperms. So that will trigger the immune reactions, autoimmune diseases. So these Sertoli cells, by forming the blood testicular barrier, it protects the immune reactions, protects. Okay, immune reactions against the your own antigens. Not only that, see, I have told you, your Sertoli cells, they have an enzyme which is called as aromatase. What this aromatase is going to do? Aromatase is going to convert the testosterone from the Leydig cells into estrogens. I have told you the estrogens are going to cause the closure of the epiphyseal cell plate during the puberty. So stops the growth spurt. And not only that, estrogen in the males are also responsible for libido. Okay, males it is responsible for libido. Testosterone will be converted into estrogen. Yes, estrogen helps in the libido. Apart from that, what else? Okay, fifth one is completed. Sixth point. Your Sertoli cells produces something called as anti-Mullerian hormone. Okay, anti-Mullerian hormone. What is this anti-Mullerian hormone is going to do? See, when you are an embryo, even in male, okay, even in male, there is something called as a Mullerian duct. Okay, there is something called as Mullerian duct. Now, what is this anti-Mullerian hormone do? Anti-Mullerian hormone inhibits Mullerian duct expression. So, Mullerian duct will be regressed. Otherwise, what happens? Mullerian duct is the one which forms the uterus, fallopian tubes, cervix, uterus, fallopian tubes, cervix, right? Mullerian duct is the one which forms the female reproductive organs. But we don't want that. Actually, the Mullerian duct is there in the body. Okay, even in males it is there. So, this Sertoli cells produce Mullerian inhibiting factor, also called as anti-Mullerian hormone. It causes the regression. it causes a regression of the Mullerian duct that inhibits the formation of uterus, fallopian tubes and cervix. I have told you, Sertoli cells, what else they produces? They produces inhibin B. What is the function of inhibin B? Inhibin B down regulates FSH, important for your exam. Okay, important for your exam. So these are the important points about the Sertoli cells. Now after this Sertoli cells, let's talk a few more important points about let's say Sertoli cells, spermatogenesis is happening. Now where the sperms will come? Okay, Sertoli cells helps in the spermatogenesis. The in detail points about the spermatogenesis we'll discuss later. Now, seminiferous tubule, Sertoli cells are forming like blood-testicular barrier, spermatogenesis is happening, sperms are produced. Now where do you think the sperms will go? Sperms are going to enter into rete testis. Okay, sperms are coming where now? Rete testis. Now what is the important point about the rete testis? In the rete testis, the semen is concentrated. The semen is going to get concentrated. Means fluid will be reabsorbed. Fluid reabsorption. Okay, there will be fluid reabsorption and the semen is going to get concentrated, and this occurs with the help of estrogen. Okay, estrogens are helping in this process also. Fluid reabsorption, and that is going to cause the sperm concentration. Now from the rete testis, the sperms are going where? The sperms now from the rete testis, they will go to epididymis. So now what happens? I have told you, from the seminiferous tubule, the sperms are going into rete testis. From the rete testis, the sperms will go to the epididymis. In the epididymis, important points are there. The sperms are going to stay here for almost 2 weeks, 2 to 3 weeks. 2 to 3 weeks. Okay, 2 to 3 weeks they will stay there for almost 20-25 days they will stay there. Okay. Now, in the epididymis, sperms will be matured and they will acquire the motility. First important MCQ in your exam, sperms acquire their motility, movement for the first time where in the epididymis. So, acquire motility. Okay. Next, not only that, I have told you there will be maturation of the sperms. Sperm maturation. Okay. Sperm maturation here, what I mean means, now here at this place, now the sperms are getting the receptors. Now they are developing the receptors which can go and bind with the ovum. Okay. So sperm maturation means develops receptors on its surface, develop receptors for binding with zona pellucida of the ovum, with the zona pellucida of the ovum. So these are the important points which I want you to know. Okay. So sperm maturation will occur in the epididymis, acquires motility in the epididymis. Don't say capacitation. Capacitation is not happening here. Capacitation is a totally different thing. Okay. Capacitation occurs in the female genital tract where the sperms will become more motile. Okay, sperms are going to shed off the most of the components and they will become more hyper motile. That is a capacitation. That's a totally different thing. Maturation of the sperms, motility for of the sperms, not the hypermotility, just motility of the sperms. They will occur in the epididymis. Okay, from the epididymis, the sperms are later going to move into the vas deferens and from there to the urethra, from there to the female reproductive tract. Now, what else I have to teach you here? What else missing? Is some important points about the spermatogenesis. See, for the process of spermatogenesis, starting from the spermatogonium to the spermatozoa, how much time it will take? The duration is going to be somewhere around 74 days. You know, the spermatogenesis starts after puberty. Okay, it starts after the puberty. It takes how much time? 74 days. It takes 74 days after. Okay. Now, one spermatogonia is going to give rise to how many sperms? One spermatogonia is going to give rise to 16 sperms. If you want to know the average will be 8 to 32 sperms. Okay, so in the humans, one spermatogonia, okay, one spermatogonia is going to give rise to how many sperms? 16 sperms. Okay, these are the basic singular points which I want you to. After this, look carefully here. Let me show you the spermatogenesis step by step. Look, first what you will have, you are going to have the primordial germ cells. The primordial germ cells later will be converted into spermatogonia. I have told you where are the spermatogonia present. Spermatogonia are present in the basal compartment. Okay, they are present where? In the basal compartment. Now, from here, the spermatogonia, they will move. From here, they will move where? They will move into the abdominal compartment. Okay, from here, they are going to move into the abdominal compartment during puberty, and they will start to convert into, the spermatogonia. They will undergo mitosis and they will be converted to primary spermatocyte. Now, the primary spermatocyte will undergo meiotic divisions, meiotic 1 and meiotic 2. Okay, the primary spermatocyte initially converted to secondary spermatocyte, and secondary spermatocyte later converted into spermatids. After second meiotic division, what do you have? Spermatids. Okay, so spermatids are the round cells. Now the spermatid later it should convert into a spermatozoa with the tail. So this process, this differentiation, conversion of a spermatid into mature sperm, this is called as spermiogenesis. Okay, this process is called as what? Spermiogenesis. Okay, so this is what they will ask you in your exam. So spermiogenesis means the conversion of spermatid into spermatozoa. This is spermatid into spermatozoa. Okay, so this is called as spermiogenesis. For the spermiogenesis, you require the testosterone. Okay, who is required for this spermiogenesis? Testosterone. Now from here where it should release. See, from here the mature sperm, the spermatozoa, it should release into reedy testis, into the epididymis, into the vas deferens. So the movement, this is called as permeation. Okay, from the seminiferous tubules later it have to go into the reedy testis. This movement is called as what? Spermiation. What do you require for this spermiation also? Testosterone is required for this spermiation process. Okay, so testosterone is required where? The testosterone is required for the process of spermiogenesis, spermiation. But this initial phases, all the initial phases in the spermatogenesis, the initial phases, you require FSH, follicle-stimulating hormone, the follicle state. Okay, so follicle-stimulating hormone is going to control the activity of the Sertoli cell proliferation, Sertoli cell maturation. Okay, so initial stages of the spermatogenesis is under the control of FSH, and the later stages of the spermatogenesis, that is spermiation and spermiogenesis, they are under the control of testosterone. Estrogen already I have explained you. Estrogen is going to help you in the acrosome development, acrosome biosynthesis, and estrogen functions in the male. Estrogen helps in libido. Estrogen is going to close the epiphyseal plates, stops the growth spurt. Okay, so with this, the important points which you need to know regarding the male reproductive physiology are completed. The actual topic of the spermatogenesis step by step that I have already discussed in the other video, you can watch that video. So with this, all the important points whatever you require, okay, for your exam are completed. Hope the video is going to be helpful for you. Thank you.